Okay, then I will start. And yeah, first I also want to thank you for the opportunity to present my work here at the Micro Symposium. And I want to talk about our efforts for the design of a protein-protein interaction inhibitor that's targeting the end domain of P97. So what I will talk about, first I want to give you a short introduction on our target protein, then say what is the aim of our project, and then show first results from our non-covalent fragment screening we performed on P97, and also from some covalent efforts we do. So what is our target? Our target is the AAA ATPase P97. And P97 is an essential protein involved in numerous cellular processes and plays a multiple key role during protein homeostasis. So, for example, it is involved in different degradation processes, for example, in the endoplasmatic reticulum-associated protein degradation, the mitochondrial-associated degradation, or the chromatin-associated degradation. And there, P97 extracts ubiquitinated proteins from their origins and handles them to the proteasome system for degradation. And furthermore, P97 is also involved in numerous membrane fusion processes in the cell. So how does P97 look? It's a little bit like two stacked donuts, or a little bit more scientific spoken, we have got here a homohexamer, and one monomer is subdivided into two ATPase domains, CD1 domain and CD2 domain, and we have got an N-terminal structured terminal domain. Yes, what is P97? It is a little protein machine which converts the chemical energy which released from the hydrolysis of ATP into mechanical work. And how does it look like? So the P97 has got two conformations. If ATP is bound, the N-terminal domains are here looking out of the plane. The plane is thought here as CD1 domains. And when the hydrolysis happens, then in the ATP conformation, the N domain make a big conformational change and then lies inside the thought plane of the D1 domain. And this movement of P97 makes its mechanical force on their targets to extract them, for example. So how this protein machine is now connected to different cellular processes. So the functional diversity of P97 is mediated through the interaction with a large number of different cofactors, which mainly acts to the N domain, of P97, and some of them also go to the C-terminal part of P97. And the protein cofactors connect P97 then to their substrates, and then the process can happen. So we are interested in the N domain, so I will have a look more here a little bit in more detail. So in the N domain, we have got two major binding sites for cofactors. One lies here between the two subdomains of the N domain, where cofactors harboring a UBX or UBXL domain bound, or also with a VEM or VBM motif. And the second binding region, the SHP binding site, is here in the C-terminal part of the N domain, where cofactors harboring the so-called CHP binding motif binds. After I introduce shortly our P97, it's the question, why is P97 pharmacologically interesting? So, why we want to design inhibitors for it. So first, the wild-type form of P97 as a central component of the ubiquitin proteasome system would be an interesting target for cancer. And on the other side, mutation forms of P97 are associated to neurodegenerative diseases, like, for example, the Paget disease of the bone and frontotemporal dementia, or the familial type of the amyotrophic lateral sclerosis. So when we talk about inhibitor, what is already known about inhibitors for P97? So first, there are the most known inhibitors for P97 are competitive ATPase inhibitors, which mainly acts on the D2 domain. Then we have got some allosteric ATPase inhibitors, which inhibit the communication between the D2 and D1 ATPase function and also shut down the whole ATPase function of P97. Then we have several compounds where we do not really know where they bind or how the mechanism of the inhibition is, and we are interested in the N domain. And in terms of the N domain, there's at the moment only reported one natural product, xanthomol, von Hobbs, where a mechanism over the N domain is postulated. So we want to design a new inhibitor class for P97. And our strategy to design is a protein-protein interaction inhibitor targeting the N domain of P97, which in the end should inhibit the cofactor binding to this N domain of P97. Why we wanted to do that? Because we think that in contrast to ATPase inhibitors, this strategy could reach in a more selective way of inhibition than shut down the whole function of the P97. So our approach is a fragment-based approach to first define chemical starting points, and then, of course, synthetically grow them, and in the end, hopefully get such a protein-protein interaction inhibitor. So I want to now give a brief summary of our results we have so got far. When I started my PhD, we first make an in silico approach. So till now, there are no complex structures in the PDB that showed a small molecule or fragment with the N domain of P97. So we start with the apo structures or complex structures of known cofactors and make them first grid-based and also docking approaches to identify maybe suitable binding sites where fragment could bound. For example, here with the FT-map algorithm, we could also reproduce that the UBXL and the SHP binding site is interesting for small molecules. Besides that, we make excessive molecular dynamic simulation to get something known about what is the water behaviors in these subpockets. And we find out that, for example, for the SHP binding site, there's the explicit water network in it. And also this water network, one molecule is seen in the cofactor binding where it has got a bridged function to P97. And so we decided to include our founded water molecules into our screening approach. So based on this structural analysis, we choose the SHP binding site as most attractive target region for small molecules, which there are three interesting binding sites. First, the SHP1 binding site, where a conserved lysine of cofactors bound, and also some interactions between the backbones of the cofactor and P97 happens. And the SHP2 binding site here in yellow, where more aromatic residues of the cofactor binds. And the third site, where solvent exposed cysteine residue lies, and we think that there is maybe the possibility to make a covalent approach to fix their fragments or molecules on that. So first of all, we make a virtual screening. I want not to go into deep detail here. I only want to say that we first set up pharmacophore screening. Then the remaining fragments were docked several times. And in the last step, the binding poles were checked for their stability using different ND simulations and the best or the tight and stable bond fragments were then selected. We, for testing the approach, only bought 10 compounds and test them in STD, NMR, and also with the Biolayer interferometry method. So to these two methods, I will come later, especially to the Biolayer interferometry method, because it's not so common for fragments. From these 10 compounds, we got binding signals for 2 fragments in both assays. So these 2 fragments here showed a binding event in the STD NMR assay, and also we could get an affinity range from the fragments, which are in the typical hundreds micromolar range. After we set up these screenings and the assays, we decided to perform a bigger fragment screening using the Biolayer interferometry as primary screening method, where we screened the in-house library at the University in Bergen, which consists of over 650 fragments. And one big problem here in the start was that we have no valid positive control for the isolated N domain. So we first performed the screening with the ND1 construct, where we have got ATP as valid positive control to see if the assay works, if the screening works, and then transform the conditions to the screening with the N domain. At the end of our dose response selections, we then tested the selected or the founded hits again with the STD NMR method. So only few short sentences about the Biolayer interferometry, because maybe not everyone know of it. So what we do here is we have got an optical sensor, we immobilize our target protein, and then we send through light through it, and the light is partially reflected on an optical surface on the Biolayer surface. And the reflected light then makes the interference pattern. And if a ligand bound to our target, there is a density change or a thickness change of the sensor, of the Biolayer sensor surface. And then we detect a shift in the interference pattern, which is our signal. The BLI is not so common used as biophysical method for fragments, so the standard is more the SPR method. So the question was, is the BLI a valid method to detect fragments on P97? For that, we use the ADP as a positive control. We have got also reported SPR data in the literature, and we measured it, and we could really good produce the affinities here with the reported affinities using SPR, and also the kinetic data is in the same range of from the BLI measurements. So with this positive control, we use then as marker for the whole screening with the ND1 domain, and as the quality factor for the screening, we get a Z factor about 0.67. Everything about 0.5 says that the assay works really good. So in the ND1 screening, we could identify 14 fragments, show the dose response. And then after we transformed the assay conditions to the N domain, we got 19 fragments showing a dose response. So here are some example sensograms for maybe, for example, 12.2 and 12.7. These are close analogs which produce similar sensograms and signals. Here 12.8, which showed a biphasic fallout, and maybe have more than one binding site on the N domain, which is not untypical for fragments. And here also an example of some unspecific binding, for example, of 12.12, where in the higher concentrations, not really a saturation signal gives in the sensograms. The founded fragments then, we make an orthogonal assay with our setup STD NMR assay. From there, 3 fragments show no binding anymore. And also here one example of it. So we have got the off-resonance spectrum, and we use for the STDR effect only the aromatic parts of the ligand. Also the fragments all have aromatic groups in it, because it is far away from the protein signal. And when the ligand is bound to the protein, of course, there is magnetization transferred to the ligand, and then we got a spectrum in the difference spectrum. So till now, there was only one fragment screening reported for the P97, where they want to find new molecules or design a molecule that inhibits the D1 ATPase function. And at the side product of this screening, they found also 6 fragments bind to the end domain that we use. And to compare the reported fragments with our fragments, and we plotted the chemical space of the fragment library in Bergen and plotted the positions of the found fragment of our work and this one of Cimenti et al. Into it, and found that they all lie here in this area of the chemical space. Only 12.2 and 7, I showed already, lie here some in other chemical space, and 1 fragment from the reported fragment screening. Then we go a little bit more in detail and make ligand-based pharmacophore analysis on the fragments, and could also see that some of the fragments shared similar features. Here, one example, these fragments showed 2 aromatic functions here and a hydrogen donator function in this at this point. And 2 also shared a parasubstituted hydrophobic group. So so the question is now, are the founded fragments somehow better than this one we already know? If you look at the ligand efficiencies, that gives a feedback, fragments maybe is promising for further develop. We see that the new found fragments of our work showed a little more higher ligand efficiencies compared to the known of the literature, and lies more in the mean values of reported successful fragment-based drug discovery screens. And also our fragments are not so dominated by lipophilic effects. With here the lipophilic ligand efficiency von Aztecs is the lipophilie taken into account of a fragment, and you want to have not so high values, not so low values here if you start optimization of that fragment. So the fragment seems to be a little bit better than this one reported. So, but of course, we have only the fragments. We have to have some structural information for the further optimization. And we first to postulate the binding poses using mixed solvent MD simulations, where we simulated the end domain in a high concentration with the selected fragments. And we analyzed the MD simulations, how often a fragment during simulation could be observed in a certain region of the protein surface, and the result confirms the structural analysis of the virtual screening that most interaction occurs here in the SHP binding site, showed here in different ketones. And further, we could postulate for 4 fragments a binding pose. You see here different ligand occupancies for selected ligand atoms during the MD simulation, and that showed a defined pose. So in parallel, of course, we tried then also to crystallize the fragments, and we found then good conditions for ND1 construct of P97, and in co-crystallization experiments, we could then identify TROL2. Okay, the density is only partial, and if we have not the anomal signal of the Baum uptone, I would not trust the densities, but because we only found this very significant anomal signal of the Baum uptone only in the co-crystallized crystals with TROL2 or not in the APO structures, we checked for it. We think that this significant signal that the TROL2 bounds to this binding region. And for then one interesting thing is also that the binding region is the same binding site which is postulated by the MD simulations. To get here a little bit more confidence of the binding site, we performed then 15N HSQC NMR measurements, and also get for the TROL2 a significant signal here in the SHP2 site for the where we have got the highest chemical shift perturbation for the isoleucine 114. There were some other chemical shifts, but there maybe are only artifacts of the construct, because we used only the end domain of P97, where we got the assignment, because the ND1 construct would be too big. And these areas are normally masked from the D1 domain, so that only this region here of the SHP binding site is left in the biological relevant system. With these 15N HSQ-C measurements, we could also get signals for the other 3 fragments. For 12.7, which is close analog, we get the same chemical shifts. And also for 12.12, which also binds postulated to the SHP2 domain. The only fragment which shows a little bit discrepance was 12.8. It lies here in this region in the MD simulations. Okay, we get that here this chemical shift, but a bigger one is found more in the UBXL binding site. Because we have now valid crystallization condition, we go then further and screened a mini fragment library in X-ray fragment screening. This library consists of very small fragments, and but they are very high soluble, so that we can have soaking concentrations up to 1 molar for the crystals, and the crystals also survive at very high concentrations. So we recorded different data sets here in ESRF, also at Bothe in Berlin, and using the Panda algorithm also to identify fragments. And the work here is still in process, but we could say that we have identified 23 fragments in the densities. Most of the fragments binding here between the interface regions, between the N and the D1 domain. But in terms of the SHP binding site, the N domain, the SHP binding site is the most prominent binding site of the fragments. So only some structures we see here of the SHP1 binding site. We addressed also in the virtual screening approach, and we find here the postulated interactions here with the backbone. And there where the leucine lies from the cofactor, we have got the more hydrophilic, the aromatic systems of these fragments lying. So because the time is a little bit running, I only want to give a short overview of the covalent approach we also set up. So in the structural analysis using isolated MD simulations, we found out that there are 2 cysteines in the N domain which show a high accessible surface area, and maybe because of that are attractable for a covalent modification. Again, we selected the cysteine 1.94, which lies here in close proximity to the binding site of the cofactors. And the idea is here to find something that bounds selectively to this cysteine and maybe help us to get a tight binder here in this area. Also here, we may got a in silico approach first with a covalent docking approach and also MD simulations. And we choose that fragments that show in the MD simulations a selective bound to the cysteine. We measured here the distance of the warhead to the our target cysteine, and then performed a mass spec analysis. And from also we ordered first only 10 compounds for testing. We found 1 compound here, 1 fragment with selectively bound only to the cysteine 184. So I want to go to the summary. Of course, now the question is, what is the relevance of the identified fragments in terms of the design of a protein-protein interaction inhibitor? And you see here in black the crystal structure of a cofactor, UFD1, which binds here to the SHP binding site. And we could identify with the different approaches fragments bounding here in this area where the conserved lysine binds. Also aromatic fragments, making the interactions of the aromatic residues of the UFD1. And as an additional anchor point, we have got here cysteine 184. And the idea is, of course, now to link and grow the fragments to a more potent and affine inhibitor, which then in the end could inhibit the interactions of the cofactors to the SHP1. Of course, from the crystallographic screening, the fragments are very small. And but with this new crystallographic data, of course, we could now fine-tune our virtual screening approaches and then make a new virtual screening with bigger libraries, because now we could train our models on experimental data we have not had at the beginning of the project. So, and with this, I'm at the end and want to thank for the cooperation partners, for the university in Bergen, where we could perform the fragment screening, for the university in Jena, which helped us with the 15N measurements, and all other groups, parts of the Schindeling group. And thank you for your, for your interesting.